There are some people that said when we start coming up with this, serotonin, that's old hat. We don't want to do serotonin in psychiatry anymore. We want to do ketamine or glutamate or some other novel mechanism. So there was hostility towards concept because it was serotonin based. That was the negative attitude. Then there was the positive attitude that was typically for treating psychiatrists or researchers that were treating psychiatrists, because they understand that the most important thing for the patient is not mechanistic novelty, it's clinical novelty.
Welcome to Power to the Patients. My name is Brandon Lee. I'm your host. And today I'm joined by Dr. Jacobsen, a neuroscientist, inventor, and CEO of Evecxia Therapeutics. So excited to be having this conversation, Jacob.
Thank you for having me today. I'm really looking forward to it.
Yeah, of course. Hey, I'm very excited to jump in, but maybe just give me a brief overview of your background and how you started working on this really cool work.
Well, I have sort of a special background. I'm originally from Copenhagen, Denmark, hence a funny accent. And I actually did my master's and my PhD training together with a Danish pharmaceutical company that's called Lundbeck Pharmaceuticals, that are exclusively CNS focused. So that was, I did most of my training and of course been exposed to a lot early on in terms of how you develop drugs and the things that are in drug development that is beyond the science per se. So that's sort of my background. And I also spent about a decade at Duke University, mostly working on the serotonin synthesis amplification project that we then spun out as Evecxia in 2019. So essentially I have sort of a broad academic, pharma, biotech background focused around the brain, focused around mechanisms of action of drugs and drug development and drug discovery. And in terms of how we came up with serotonin synthesis amplification, so we didn't really come up with it because this is an idea that in other names has been kicking around all the way back to the 60s and 70s. And what happened was that many years ago, I found out that by playing around in the lab, that the precursor of serotonin 5-HTP was phenomenally potent in augmenting the serotonergic effect of serotonin reuptake inhibitors, both in terms of actually looking at serotonin release in the brain of mice, but also some functional behavioral outcomes. And then I started reading about 5-HTP and I realized there was a tremendous amount of clinical data out there indicating that 5-HTP could augment the antidepressant effect of serotonin reuptake inhibitors, which of course piqued my interest because it has been known for now quite a while, the last several decades, that SSRIs, for instance, they work, but they're only modestly effective. So there's been this big push to develop drugs that you can add on to an SSRI, for instance, to augment the efficacy. And what I also found that, somewhat to my surprise, was that the safety record of 5-HTP in small clinical studies were surprisingly good, even though there were more than 150 published studies on 5-HTP from the 60s, 70s, 80s, 90s, all the way up till today. It was also interesting because safety is such an important thing for psychiatric drugs, but then I also found out the 5-HTP had an absolutely lousy PK profile that makes 5-HTP as a naked molecule completely inappropriate as a drug product. 5-HTP only have a half-life of around 2 to 3 hours, so you have to take it 4 to 6 times a day to get reasonably sustained exposure in the blood, which of course is completely impractical. And it also looked like 5-HTP had poor bioavailability, which we subsequently have found in Evecxia is only about 5%. So 5% bioavailability, half-life 2 to 3 hours, you cannot be a drug. But then I thought, what about if you could come up with a technology that could prolong the action of 5-HTP and hopefully also increase the bioavailability? Maybe you would get a practical drug. And I probably had those thoughts because given my background in pharma and biotech, that there was a big focus at the time when I was in pharma biotech to get compounds into development that had appropriate PK. Because there's so many drugs that never made it, probably because you didn't get enough constant exposure. So I was aware about all these factors. Then at Duke, we did some studies where we tried to emulate sustained release delivery by giving the 5-HTP in the food. So the animals would get it over a significant amount of time. And also played around with trying to give co-administration with various doses of a compound called carbidopa that can enhance the bioavailability of 5-HTP. And the key findings here were if you get 5-HTP as sustained release, so to speak, you get dramatically better tolerability. And also we found we can get these sky-high levels of 5-HTP into the animals, and it could very potently boost serotonin in the brain, but the animal looked quite fine. It was interesting on the academic setting because it jived well with what has been reported in the literature. 5-HTP as a compound is probably very well accepted for the body. And then we also found that you could combine 5-HTP with this carbidopa compound. But if you gave the carbidopa at tremendously low doses, but you gave it as sustained release in close spatial and temporal proximity with 5-HTP, you could still multiply 5-HTP's bioavailability manifold, but without the carbidopa ever showing up in the bloodstream. So this is a local effect by the carbidopa in the intestine. And then eventually when we started Evecxia, sort of as a side note, then we actually found that the same effect could be shown in humans and were even more potent in humans. So that was a kind of a case of successful pharmacological translation from animals to humans. So that is a little bit about the background where this whole thing comes from. There's more to it.
Tell me a little bit more about what it has been like fighting for this mechanism of action.
It's been very interesting because the waters are very divided. There are some people that said when we start coming up with this, serotonin, that's old hat. We don't want to do serotonin in psychiatry anymore. We want to do ketamine or glutamate or some other novel mechanism. So there was a hostility towards concept because it was serotonin based. That was the negative attitude. Then there was the positive attitude that was typically for treating psychiatrists or researchers that were treating psychiatrists because they understand that the most important thing for the patient is not mechanistic novelty, it's clinical novelty. Essentially, if you're a patient and somebody can give you a drug that all of a sudden you respond better, that's all that matters to you. That's all that matters to the patient, clinical novelty, to the patient, to the physician, to the payer, and also to society. Mechanism, in some respect, irrelevant, even though, of course, novel mechanism also overlaps with the clinical novelty, but is sort of a Venn diagram. So it's been very divided, but there have always been strong supporters. And I remember at Duke, there was 2 chairs of psychiatry at Duke in a row that was very supportive of this whole idea because they think, this is probably going to work. This is sort of shovel ready. But we had quite a few problems getting grants for it. We got some. So it's sort of a combination of we don't like this because this is not fancy, and we like this because this is probably going to work.
How did you fight through the early days of? I can only imagine that you have to fight a little bit of an uphill battle before things really take off.
When I started working full time at this project at Duke, I've always worked. First, I did something else at Duke as a postdoc, and then sort of doing this on the side, the 5-HTP sustained release. And then I started working on it full time for 2011. And I thought, okay, I'm going to give it a couple of years and see whether we can get this off the ground. It ended up taking 8 years before we finally spun out the company from Duke. We did get some early seed funding in the beginning of 2016. And we started sort of the early parts, proto version of Evecxia. But then we got millions of dollars, a few million in 2019 and could start the development. So the point is that it always kept on going up. It has always been going up, but most of the time it's been going up a lot slower than I wanted it to.
That's always the case, I think. 8 years is tremendous persistence. What were you seeing in that time that gave you the confidence that, this is the right path and it was worth persisting versus not?
I have all the time, I've been thinking in Jews, I was alluding to before, the clinical novelty, because that's really the only thing that matters. And if you look at all the literature, so the whole idea about boosting serotonin synthesis to treat patients better has been around since, as I mentioned, since 70s. So back in the 70s, you would have quite commonly that either 5-HTP were given on top of, back in the day, would be a tricyclic antidepressant. but those drugs work the same way as an SSRI, blocking the serotonin transporter. Or it would be tryptophan. And of course, tryptophan mostly becomes other things than serotonin, but that has also been used. And back in the day, actually also lithium has been used a lot as an adjunctive until around the 90s, until the advent of the atypical antipsychotics. And most of the preclinical and clinical data suggests that adjunctive lithium in depression works by boosting serotonin synthesis, which is rather non-controversial. It's in the literature. More recently, as you might know, about a decade ago, there was some interest in methylfolate as an adjunctive in depression, and it works a little bit. So methylfolate is a cofactor in serotonin synthesis, and methylfolate is not particularly potent as an adjunctive antidepressant, which is to be expected because giving a cofactor in synthesis is just not a very potent way of enhancing synthesis. So there's all this clinical data that directly points to if you can boost serotonin function beyond the SSRI effect, you treat more patients better. And then, of course, what has been found, this has sort of been a couple of things about the serotonin transporter and humans. It has been known for at least the last 25 years, based on post-mortem studies and later brain imaging studies, that the human forebrain has very low levels of serotonin transporters, about 1/50th of what you can find in mice, which is sort of interesting. So that means that the therapeutic target of the SSRIs is a low prevalence protein in the brain. So that fits really well with the SSRIs. Yeah, they work, but most people, they don't work particularly well. And at the same time, it is also well described that the SSRIs, to get any therapeutic effect out of SSRIs, you have to block almost all of the serotonin transporters. Again, this is non-controversial. This has been published in numerous times. And the interesting thing is, and this actually, a lot of this data came out after that I'm going to talk about now, that I started being interested in serotonin synthesis amplification. It's actually been found using PET studies that SSRIs only elevate serotonin modestly, inconsistently, and with a delay in the human forebrain. There's not a lot of data on this. So this is PET data, positron electron tomography imaging. But the data that's available, which is a handful of studies, they all point to that way. And the interesting thing is also that, as a side remark here, that acutely an SSRI will not increase serotonin release in the forebrain. Acutely an SSRI will either do nothing or actually decrease serotonin release and serotonin function in the forebrain. But it's interesting because a lot of patients that start taking an SSRI, they get worse before they get better. So the pharmacology of SSRIs and the neurobiology and the clinical picture completely jives. And all this points to that if you can boost serotonin beyond the SSRI effect, you can probably treat more patients, which all this data over the last 50 years involving at least 30 positive studies using 4 or 5 different compounds point to. So this is really why I keep going, because I think there's a high probability that serotonin synthesis amplification can be a broad spectrum and very safe and highly scalable approach to augment antidepressant efficacy when an SSRI on SSRI is insufficient.
I love your kind of framing here around clinical novelty instead of like mechanistic novelty. Can you break down what are the kind of key components of clinical novelty you're most excited about being able to show here?
I think that safety and scalability is important because you have to remember that there's a massive shortage of mental health care in the US. Less than 20% of US psychiatrists take new patients. There's a shortage of reimbursement as well. So it's very exciting with more complicated treatments such as TMS, quite effective treatment for TRD, treatment-resistant depression. The issue is there's so many patients out there. So it's a very small proportion of patients that will ever get access to TMS. And you've also seen, for instance, that we're very excited, everybody, about the Spravato ketamines. Spravato has so far, in 6 years, been dosed to around 100,000 patients. So currently in the US, there's probably 5 to 6 million TRD patients. So this is a tiny minority of the sickest, probably best insured, and patients that are living close to major population centers. Most patients are not like that. So if you really want to make an impact at scale in, for instance, depression, OCD, anxiety, you need to have more economical and scalable treatments. And EVX-101 could be those.
What makes this more scalable than Spravato? I think certainly TMS of course, quite limited by the hardware, let's say. But how do you think about that versus like Spravato?
Well, Spravato, just a drug, costs $30,000 a year. Okay, so that's a high cost, and you have to go to the clinic. The clinic also costs tons of money. I think, okay, don't hold me up with this, but I think I've been told by a treating physician from Yale that is about $6,000 per instance, the total cost. That's just a lot of money. Somebody's going to pay for that. Who's going to pay for that, Brandon? Is like the American Society going to pay for that? Like, is Medicare going to pay for that? Like who's going to pay? Okay. So if you have a tablet, you can take it home. That's highly scalable. And if it's safe, that also means being given up by a much broader segment of care providers and also more patients. Let me give you an example. So we all know the SSRIs, Prozac. They got on the market in the late 80s. And in the end of the 90s, you had 4 SSRIs at the same time that were blockbusters. And you had all these books coming out of all these people that have their life changed to the better of SSRIs. But you know what? SSRIs, they don't work any better or any differently than the tricyclic antidepressant from the 1950s. What's the difference between the SSRIs and the tricyclics? The SSRIs, they're more selective on the target, the serotonin transporter, and they're safer and better tolerable. So the SSRIs is purely a safety play. It's not an efficacy play. But you have a better safety and better tolerability. More physicians can give it out and can be given to many more patients. So that's where you get the impact. And I'm old enough, Brandon, to remember in the 90s when everybody was talking about that the falling suicide rate, essentially globally, was to a large extent connected with the implementation of the SSRIs. Then, of course, in the US, the suicide rate turned and started going up again in the 2000s, but that may be a different story.
Given the kind of scalability play here, I'm curious how you think about going after adjunctive, it's not quite treatment resistant, but like insufficient response to SSIs, as opposed to just going for your like straightforward scalable play out of the gate.
You can't displace, like simply from a healthcare economic standpoint, you cannot displace the SSIs. You're not, and I don't think you should actually, because you have to remember, so right now people, it's like very popular, particularly among the psychedelics people, to bash the SSIs, these terrible, terrible, terrible drugs. But the fact is that they're very safe. They work really well in about 1/3. And then there's an extra third on top of that, they get a measurable response, and most people actually tolerate the SSIs. So the SSIs are going to keep on being first line, and that makes sense. And also from a payer perspective and a healthcare perspective, generic Lexapro or generic Prozac is like 5 cents per day. Yeah. So you want to start with that. And right now, the payers typically require that you try 2 SSIs before you can try something next line that is potentially branded. So it's going to be impossible. And also from, I think, really a treatment perspective, it makes sense to start with an SSI as of current. Because of this fact that most patients actually tolerate the SSI, but also most patients that take an SSI have an incomplete response. So you have this massive middle group in depression patients, OCD patients, anxiety patients, bulimia, other indications that are partial or little bit responders to SSIs. So that's the massive market segment you can address there, because people stay on the SSI. So that's why we're going for adjunctive. And you have also seen that the vast majority of drugs that have been development in the psych space or the depression space over the last 20 years has been adjunctive. Almost, I think actually all of the drugs that have been developed in OCD, I might be missing something over the corner, but like 90% of the programs have been adjunctive. And that is because in OCD that most patients actually get something out of it that is measurable, but it's a small minority that get a complete response. So particularly in OCD, you have these like millions of patients that are long-term on an SSI. So that's why we're going for adjunctive.
Yeah, makes sense. So maybe going back to like the kind of clinical novelty framework, we had, a category of things that are beneficial here from a safety and a scalability perspective, but we haven't spoken about efficacy yet. How should I be thinking about efficacy here?
Well, that's a good question, Brandon, because we really don't know. It will have to be seen, doesn't it? Because we are hoping to start phase 2 next year. We have successfully completed phase 1 and non-clinical talks with our lead drug, EVX-101, and with essentially clean safety, both on the clinical front, but also on the non-clinical tox front. And so since there's never been an effective drug to cause serotonin synthesis amplification in a clean fashion, it will have to be seen. So the phase 2 is going to, I think, be very telling. But given that people, like researchers, has been able to, even with the whatever 5-HTP that's available and just random doses, being able to repeatedly report that you can augment antidepressant efficacy in depression mostly, but also some studies in OCD, some anxiety. That's very promising, that you even with non-optimized doses, dose levels, dosage forms, and paradigms can get an effect that indicates that there's reason to optimism.
Certainly very exciting. Is it too soon to talk about how you're thinking about that study and designing it?
It's very simple, really. The nice thing about adjunctive studies is that this has been done so many times before. So initially, our key indication is OCD. And OCD is interesting because there are more OCD patients than schizophrenia patients. And in OCD, half of the patients are severely impaired. You probably don't meet them much out in the society because they sit at home a lot of the time. So OCD, there's no next-line therapies approved. It's only the SSRIs. But the study design is really plain vanilla. So you enroll OCD patients that are taking an SSRI at an approved dose by the FDA and can be verified to taking that SSRI, and then you randomize them to either adjunctive placebo or adjunctive EVX-101 for 10 weeks. And there's really only one instrument to rate the OCD that has been used and has been accepted, and that is the Yale Brown Obsessive Compulsive Scale, YBOX, developed originally by Wayne Goodman back in the 80s.
It's interesting that you're going for OCD first. Talk to me about that decision.
Well, because think about it. Depression, adjunctive therapy. You have multiple generic incumbents. You have aripiprazole, Abilify is generic. Seroquel, quetiapine is generic. Rexulti is going generic. You have several other adjunctive treatments that are approved, such as several other of the atypical antipsychotics. Most recently, you had Caplyta, which is sort of almost a fourth generation, more safe atypical antipsychotic that was approved for depression. You also have something like vortioxetine, Brintellix, Novelty, dextromethorphan/bupropion from Axone. Those are kind of switch therapies you switch to. And then, of course, you have a very rich pipeline of predominantly psychedelics in the depression space. So the depression cake is very big. There's also a lot of slices of depression cake that has been given away already. In OCD, the next line therapy cake, so to speak, nobody has claimed that from an FDA-approved standpoint. The pipeline is relatively slim. We believe that we are ahead in the pipeline. So if we get to the next line therapy space in OCD first, we could essentially own that space. So we get a much bigger slice of somewhat smaller cake, but that could overall become a bigger commercial opportunity. And then there's another thing that's really interesting about OCD is that the trials, and this is well described in the literature, are generally risk mitigated compared to depression. And that is because in OCD trials, they have way lower placebo response than depression, about 60% lower. Some of the original registration trials with OCD from back in the 90s with Prozac, there was no placebo response. Like none. Maybe I should talk about what I do know and not what I don't know. But the speculation is that OCD is just more severe disease and is more pervasive. Like it's different than normal life. We all get times in our lives where we're depressed. Most of us do not get times in our lives when we have OCD. So this is a discrete entity. And also OCD is much more constant, as all diseases do, but it fluctuates with a less amplitude and over a longer time period. While, as you probably know, depression can really go up and down from, month to month, week to week. So that might be part of the explanation.
Yeah, the episodic nature here is kind of interesting.
Yeah, it's interesting, but, and also the OCD patients typically come with a long history. So you're much less likely to get inappropriate patients in your study because you can get access to the medical records and they've been sick for a long time, particularly if it's adjunctive therapy. And then finally, this is maybe a little bit anecdotal, but I've heard that from OCD KOLs. I've also heard that from OCD patients themselves, that they're really good at taking medications.
Oh, interesting. By nature of the kind of condition, adherence might be higher.
That is the thing. So that's why we're going after OCD.
Yeah, certainly. And, I don't know if depression will be an exclusion for this program, but we know that OCD is often an exclusion for the depression program. So there are folks who are primed and potentially interested in research that's, at least we see on our platform, that's we're looking for a good place to connect them with.
They might be able to enroll in our study.
Potentially a good overlap there. Hey, I want to change course a little bit here. I've heard you describe animal models as famously ineffective in psychiatry. Why do you hate animal models?
I love animal models, but I dislike specific part of animals. I think we should use things what they can be used for and not use things what they cannot be used for. So the thing is, of all the psychiatric medications that are on the market today in the US, all of them, 100%, are based in primary clinical evidence. All drugs that have been developed in psychiatry based on primary efficacy in animal behavioral models of psychiatric disease has failed. So it hasn't worked. We've been doing this now since the late 70s, since the introduction of the forced swim test in 77, 78. These methods, they don't work to find novel mechanism drugs. They don't look like depression. They're not predictive. They're not sensitive. They're not selective. Most of the time, you can't get an SSRI to work in forced swim test or tail suspension unless you give massive doses of SSRIs, which people typically do, maybe because they don't know any better. When you give an SSRI at a high dose, you hit a ton of different targets. So if you see, for instance, fluoxetine, the way fluoxetine is being used in the academic setting, people typically use like 10 milligrams per day or 20 milligrams per day, and very often give it like IP. So you get this bolus into the belly. So you get this massive spike of fluoxetine. So you get, and this has been described, is well known in the literature, that if you get up in the micromolar level in the blood or the brain of fluoxetine, fluoxetine hits a lot of different targets. In addition to the serotonin transporter, his other monoamine transporter, his some serotonin receptors. It hits potassium channels. It hits sigma. It hits, you name it. Okay? So the point is that, these models, they typically don't even respond to the first line antidepressant. And problem about using these models, Brandon, is it's incredibly wasteful. It causes unnecessary attrition. So let me give you some recent examples. So there have been some studies that found that R-ketamine, so the R in enantiomer of ketamine, worked really well in some animal models of depression without seeming having like hallucinogenic effects. And then because of those animal data, R-ketamine was put into a depression trial and failed. Okay. So more reason, there was a mTOR modulator in development that worked fantastically in some animal models. Nice paper in Journal of Clinical Investigation. It's like everything hangs together. It's beautiful. You put it into people in a randomized controlled trial, and there's just like no effect. Okay, I can go on and on and on and on and on. So the fact of the matter is that these models have never helped patients. And if you've been doing something, now you're spending billions of dollars over half a century, and no patients have ever really been helped. Should you continue to do that? I don't think you should, because you're diverting money, you're diverting brains, you're diverting attention, you confuse yourself, and who is suffering in the end? The patient is. Society is suffering.
Certainly a compelling case that, the kind of predictive power of these models are not great for efficacy. But where do we go instead?
We study patients, we study depression. So I don't have the answer necessarily. But what you did back in the day, for instance, people were experimenting around and observing patients. That is the way why we have the drugs we have. Think about it, for instance, the MAO inhibitors, they were an observation in tuberculosis patients in the 50s. So nowadays we have all the various types of brain stimulation modalities. Where does that come from? That comes from ECT, electroshock. Where does electroshock come from? That comes from shock therapies using chemical shockers back in the 30s. Where does that come from? That comes from the observation that epileptic attacks in some psychiatric patients could actually improve their mood. So this was sort of a serendipitous observation. But just because I don't have the answer what you should do instead of animal models, I think we should stop doing what doesn't work. Just because we cannot come up with a better model, better approach, we shouldn't keep on doing what doesn't work, right? So we should start figuring out what to do next. Allocate these resources to study patients, to start hacking away at the actual problem. So see models of depression, for instance, Brandon, they are just kind of infusing and confounding the whole field. Because then people are going to see then you'll see these references, but then these drugs, they do something in animals. And then we saw a correlation between there's some spine or something was going up in an area of the brain we like in a mouse. And that corresponded to that when you take a mouse by its tail, it dangles more. And you say, this is the mechanism. Well, these models have not been predictive to predict new antidepressants. So why should they be predictive for that? So that's the problem. I think we're confusing ourselves. And I just gave you two very recent examples, the R-ketamine and this mTOR modulator compound, where millions, tens of millions of dollars have been spent on getting negative trials. What happened when we get negative trials in depression, Brandon? Investors and pharma get skittish. Okay? That's actually, I'm old enough, so I'm Generation X. So I'm old enough to remember when most of pharma exited psychiatry around 15, 20 years ago. And one of the stated reasons at the time was, we can't do this because the animal models, they're not predictive. And I was writing about that. So now there's a new generation of people that have sort of forgotten about this. So I predict, Brandon, that we, in addition to the couple of examples, there's more, that a reason we're going to see more failures in the depression space because people are going into humans based on these animal depression models. And then what's going to happen is that then the blame is going to go, it's just difficult to do a depression trial. Well, if you know how to do a depression trial, it's actually maybe not that difficult because if you know what you're doing and you have a good drug, it's going to work. Okay? So look, for instance, at development of Abilify. There's an atypical antidepressant. Abilify works as aripiprazole. That works modestly effective as an adjunctive. And Abilify got on the market after 2 trials that was executed by very good people, had the money behind it, and was executed with care. Both trials were positive. Abilify got on the market, and Abilify has made a difference for many patients in the US and beyond.
Are we asking too much of these animal models?
Oh, we totally are. We're asking something they cannot answer us to, right? But just think about it. So I've worked with these models, okay? So when you're working, for instance, with, let's say, forced swim test, as I mentioned, and this is even in the literature, most of the time you can't get forced swim test to work with an SSRI. Unless you give some kind of unrealistic astronomical dose. Most of the studies with SSRIs and forced swim tests are using these like massive doses that give like exposure that is way non-clinical, way supraclinical. You have no idea what you're hitting, okay? People don't necessarily know that because a lot of preclinical researchers, they don't really have this idea that you have to sort of match the human plasma levels to really study the effect of these drugs in animals. So, and I've seen that. And another thing is that it kind of also depends on the strain of mice, Brandon, the age of the mice, what effect you're getting. And I remember there was like a decade or 2 ago, there was a paper published where researchers had taken, I think, 30 different strains of mice and done tail suspension. That's one of these antidepressant-like tests. And in tail suspension, what you do, you take a mouse and you hang it by its tail, okay? If the mouse is wriggling vigorously, then it's antidepressed. If the mouse is just hanging there, then it's depressed. Okay? So I think for people out in society, they would think this is sort of strange because, for starters, humans don't have tails. So already there, the translations fall apart. But anyway, in this particular study, they're taking some kind of sky-high standard dose of fluoxetine and injected it to these 30 different mouse strains, and they find that approximately 1/3 of the mouse strains, they sort of jiggled more in the tail suspension test. There was a big middle group that nothing happened, and then there were some mice that jiggled less. Okay? So depending on the mouse strain, fluoxetine can be antidepressant-like It can be depression-like, or it can be inactive. But let me give you another story, and you're going to love this, Brandon. This is actually my favorite story about antidepressant-like models in rodents. Okay, so in 2005, there came out a rodent paper in one of the best journals in psychiatry, Biological Psychiatry, Greibel et al., 2005, of this new compound that was called rimonabant, that had a clear antidepressant profile in 4 swim tests, chronic mild stress, some anxiety models, and I think they also had some biological outcome. Rimonabant, I should mention also, at the same time, other people found something similar with this compound. So this was sort of a putative antidepressant. So rimonabant was actually development for treating obesity. Got on the market in Europe for treating obesity and was withdrawn after about 6 to 9 months from the market because of severe psychiatric side effects. Rimonabant doubled the incidence of depression, doubled the incidence of anxiety, and quadrupled the incidence of suicidal ideation. So you have this drug that is an antidepressant in rodent models of depression, but is a pro-depressant in humans. Okay, so much for translatability.
The catch-22 here is that we need some filtering mechanism before going into humans, presumably, in order to concentrate investment or make sure we're making kind of like the best use of the capacity that we have to do research, right? I know. I see you shaking your head, so I'm curious what you think.
So I don't have the answer, but why would you use a filtering mechanism that doesn't work? You're not filtering anything. And you might actually be filtering away drugs that could be effective. That's what you have to think about. There might actually be out on the shelves in drug companies, there might be potential antidepressants sitting there that never got into trials because that they didn't make the mice swim more or made the mice chicken more when you hung them by the tails. So we have to do something here. But you can see, for instance, with the psychedelics are based on, clinical findings back from the 50s. And then what people do, so there's a clinical rationale, and that's what the psychedelics are. This is a serendipitous finding that is 7 to 80 years old, that then has been taken up again and made more practical. I think what you have to do, you have to start understanding these diseases a little bit better and to have the courage to go ahead and do biomarker studies, or that's kind of tough, or do small clinical trials. And actually, so the thing about that, the animal models don't work. So this was sort of up in the field 10 years ago, and NIH had some programs where they were going straight to humans. They're bypassing animal models. And now so they kind of regress back to animal models to a certain extent. So I think you just need to think about based on the knowledge we have about psychiatric pathology, what circuits, what pharmacologies might work. And then we have to shoot, because you're gaining absolutely nothing for using these animal models.
No information and potentially the wrong false negatives.
Exactly. So, but then another thing is, Brandon, so if you come and if people believe that some drug is going to work in one of these models, they can probably get it to work. Because remember what, for instance, 4 swim test is. This is movement. Okay? Movement. So there's many different kinds of movement. There's there's swimming. There's sort of just moving around a little bit because you're kind of jittery. Or there's shaking. So depending on how you're scoring your activity in the 4 swim test, you can score it different ways and get different outcomes. And of course, nowadays you use imaging software, so you can adjust the imaging software. And then, of course, adjust the dose. So a lot of things are going to start being seizurogenic or sub-seizurogenic at high enough doses. And of course, if you get you're activating the brain and the animal get jittery, they're going to have some movement, and that's like antidepressant-like, or not really, but that's what you can get. So you can sort of game the system to an extent. And then of course, we have the whole thing about academic reproducibility or not. That's another story.
Well, Jacob, this has been a fascinating and fun conversation. Is there anything that we haven't talked about today that you really wanted for us to cover?
Well, I think it's very positive. There's so much interest in psychiatry again. And I think we should note that some of the successes that have been out there recently, for instance, are all based on primary clinical data. So, for instance, you have Cobenfy for schizophrenia. This is a serendipitous observation that xanomeline could treat psychosis in Alzheimer's patients back in the 90s. You have the rise of the psychedelics. Again, this is based on an idea from the 50s that has now been made more practical and can be implementable. So I think all these treatments are going to make a difference. And same thing with the brain stimulation approaches that are based on essentially electroshock. So I think that's very positive. And so I think and these successes, I think is going to spur continued investment in the field. However, I am cautioning the field. They really have to be careful and not hampering this progress we've seen by the inappropriate use of these behavioral animal models for either depression, anxiety, or schizophrenia, because I'm fearful, Brandon, that we can see the same exodus of pharma and investors from psychiatry due to inappropriate programs that should never have been in clinical trial, but they are based on animal data that's not predictive. So I think there's a lot of hope, but we have to be careful that we don't repeat the failures of the past.
Yeah, certainly. And as you've alluded to, a large pipeline in mood disorders today, and waiting to see some successes come out of it to continue, I think, inspiring investment in more research here, which is what we all want.
Totally agree. And I hope it's going to be like that.
Yeah. Well, Jacob, again, thank you so much for taking the time. I have so enjoyed this conversation.
Dito, thank you, Brandon. Really appreciate it.